This is how to draw DNA. So we're going to start with a nucleotide as always. We're going to draw a pentose sugar. This time the pentose sugar will be deoxyribose. We're going to bring carbon 1 across to a nitrogen base, and carbon 5 as always will go up to a phosphate group. We're going to model that directly below. If they don't tell us how many nucleotides to draw, then 4 is a good number, and that would allow for 2 base pairs, so you can actually show complementary base pairing. 5th carbon up to the phosphate group, and then you want to make sure that that phosphate group can then link to carbon 3 across there. To do the second complementary strand, we're going to turn our piece of paper around. So this time I'm going to start with the nitrogen containing bases, so they're on a line. Same size rectangles, no need to make purines and pyrimidines any different. And then it means that I can draw my pentagons pointing up again because my piece of paper is upside down. Same again underneath. And across, so always carbon 1 to my nitrogen containing base, a little bit off there, but never mind. Up we go to a phosphate group, up we go to a phosphate group, connect that phosphate group back to our carbon 3 there. Then I can turn it the right way round again, and I can start to label. I could, for example, have adenine to thymine, cytosine to guanine. I showed my hydrogen bonds with dotted or dashed lines, 2 bonds between adenine and thymine, 3 bonds between cytosine and guanine. And it doesn't have to be the most beautiful diagram, you need to make sure that bonds are going to the right places, label lines are touching, and so on. Now what can we label here? We can label a nitrogen base. I'll move that along. We can label our deoxyribose this time. And we can label our phosphate group. Okay, we can label a nucleotide again, like so. Ideally we'd be using a ruler for our label lines, but it's not completely necessary, and you certainly don't want to use a ruler for the diagram. Time is going to be of the essence if you're doing it in exams. Here I can show a hydrogen bond. I'm going to write the full name, hydrogen bond. And I can show a covalent bond going between the 2 nucleotides. So covalent bond. I've got my nucleotide labelled already. One more thing for marks I need to do is actually showing that adenine is full name, thymine, full name, cytosine, full name, guanine, full name. Of course, I could have drawn my base pairs big enough to write this clearly inside, but I want things to be big enough for the examiner to read, especially as they would be reading like a copy of yours on screen. One more thing that you can label, especially if you're a higher level student, is the directionality. So up here there's carbon 5, so this is what we call the 5' end. This means that here's carbon 3, that's the 3' end. And then I can do the opposite on this side, where this is now the 3' end and this is the 5' end. So overall, my 2 strands are anti-parallel. That's how to draw DNA.